All right, so we just talked about DNA structure, how scientists figured out what it looks like and how it works. But now we need to move on to how does DNA make copies of itself and how does it pass on this information to make RNA and proteins. So we're going to start with DNA replication. And this is just the process of making an exact copy of DNA before a cell divides. Without accurate replication, cells would not be able to pass on the genetic instructions. So replication does happen in a very precise and organized way, and it follows what we call the semi-conservative model, which means that each new DNA molecule consists of one old strand and one new strand. And this just makes sure that we have accuracy while allowing for the cell to build new DNA without having to start from scratch. So the process is going to involve several enzymes. I would kind of recognize what the function of these enzymes are. And it's all going to begin with an enzyme called DNA helicase. And this enzyme is responsible for unwinding the DNA double helix and breaking those hydrogen bonds between the base pairs, between the A and T and the G and C. When it breaks these hydrogen bonds, it's going to expose the two strands and it's going to create something called the replication fork. So here's the replication fork here. It's just a Y-shaped region where the DNA is being split and copied. As those strands separate, there are some proteins called single-stranded binding proteins, or SSBs, that are going to attach to the exposed DNA to keep them from basically binding back together. There's also another enzyme called topoisomerase that works ahead of the fork to help relieve tension in the DNA that's caused by the unwinding. So next we actually have to go in and build the DNA. So I want to point out something on this image. So if you notice, here's our DNA double helix if it was not in a helical form. If you notice, here is one strand. This end is labeled the 5' end, and this end is labeled the 3' end. While on this strand, this end is the 3' end, and this one is the 5' end. What this is telling us is that the DNA strands are directional, and not only are they directional, they are anti-parallel to each other, meaning that they're running in opposite directions. So this is important because our next enzyme, which is called DNA polymerase, and that's our main enzyme that's going to go in and add those nucleotides, it can only work in one direction, and that's from the 5' to the 3' direction. So that means that one strand, which we refer to as the leading strand, is copied continuously as the DNA unwinds. So here's our DNA polymerase. Again, it can only work in the 5' to 3' direction. So as it is adding nucleotides, we can make a copy right here, a continuous copy. So this is our leading strand because it runs in a 5' to 3' direction, which is how our DNA polymerase enzyme is adding nucleotides. But on the other hand, we have the lagging strand, and this is a little bit more complicated because it runs in the opposite direction, the 3' to 5' direction. So here's our lagging strand down here. DNA polymerase is going to have to work backwards in small chunks, and it's going to add nucleotides in these little fragments that are referred to as Okazaki fragments. So here's a fragment here, here's another fragment here, fragment, so on and so forth. Eventually, these fragments are going to be linked together, and that's by an enzyme called DNA ligase. I also want to point out that before the polymerase can get started, there is another enzyme called a primase that's going to come in and lay down a short RNA primer. This is basically a starting block that tells DNA polymerase where it needs to begin. Each of those Okazaki fragments on the lagging strand does require its own primer. So once the nucleotides are added, DNA polymerase is capable of proofreading its work and capable of correcting any mistakes. DNA ligase will come in, seal any nicks or breaks in that sugar phosphate backbone, making the new strands complete and continuous, both the leading and the lagging strand. So that by the end of this process, we have two identical DNA molecules, each with one original strand and one new strand. So that's basically the semi-conservative model that we're trying to emphasize. So now keep in mind that we're eukaryotes. So our DNA isn't just floating around in a small loop like it is in bacteria. Instead, it's packed into multiple chromosomes and a nucleus. So instead of starting replication at just one point, eukaryotic cells begin replication at many origins along the DNA, and these form what are called replication bubbles, which spread outwards in both directions until they meet and the whole chromosome is copied. So let's move on to the next step, and that's going to be RNA. So DNA may be the instruction manual, but RNA is what helps carry out the instructions. So RNA stands for ribonucleic acid. Why is it called that? Because RNA uses the sugar ribose instead of deoxyribose. So essentially, we just have a different sugar than we had in DNA, and we have one different base. We have uracil instead of thymine. Another difference is that RNA is usually single-stranded and not double-stranded, and it's generally more temporary and mobile than DNA. There are 3 major types of RNA, and each of these are going to play a unique role, but they're all involved in protein production. So we have messenger RNA. This is made in the nucleus using DNA as a template, and it's going to carry the genetic message or the instructions, that's why it's called a messenger RNA, to the ribosomes, which are the organelles responsible for the site of protein synthesis. The other one that we have is transfer RNA, also made from DNA. This type of RNA is like a delivery vehicle. It's going to pick up specific amino acids and bring them to the ribosome. Each tRNA has something referred to as an anticodon that's going to match a codon on the mRNA to make sure that we are laying down the correct amino acid. The third major kind is ribosomal RNA. This is made in the nucleolus, which is in the nucleus, and it combines with some proteins to form the actual ribosomes, which again are the site of protein synthesis. So ribosomes can float freely in the cytoplasm, or they can be attached to the rough endoplasmic reticulum. So here's just a little bit of comparison between DNA and RNA. Oh, I already told you all of that. So something kind of important is this, what we call the central dogma. And the central dogma of molecular biology is we go from DNA to RNA to protein. Okay, so DNA is found in the nucleus and it stays in the nucleus, and we have to be able to get those instructions out to the cytoplasm where we have our ribosomes so that we can synthesize our proteins. So how do we get the information that's in the nucleus to the ribosomes and the cytoplasm? That's with our messenger RNA. So RNA or mRNA is produced in the nucleus. It will leave nuclear pores to travel to the cytoplasm to land on a ribosome so that we can synthesize some proteins. So if you look at the central dogma of molecular biology, notice that the whole reason we have DNA is to make proteins and to pass on our genetic information. Okay, so moving on. Way back in the early 1900s, a scientist named Sir Archibald Garrod was among the first to link genes with specific metabolic diseases. And he noticed that some inherited conditions were due to missing enzymes, and this suggested that genes somehow control protein production. So now we know that genes in DNA encode the instructions for making proteins. So when a gene is expressed, its information is transcribed into mRNA and then translated at a ribosome into a specific amino acid sequence. And that amino acid chain can then fold into a functional protein, which can then carry out some sort of task. So it can be an enzyme, which is going to help speed up a reaction, or it could be some sort of structural protein like collagen in your skin.